This video is a guide to planning investigations for Edexcel IGCSE Biology, and it'll be really helpful to those of you who are doing exams in January or the summer seasons from 2018 onwards. So we're going to be looking at this CORMMSS method. So CORMMSS will be written in all of the mark schemes. If you've seen any mark schemes so far, then you'll see that there's always a question that uses this scheme. So the CORMMSS scheme is used for questions that include the words design and investigation in them. You'll always see those three words, and so if you do, that'll set off an alarm bell ringing for you. I've got to use this CORMMSS system for answering this question. And these questions are always worth 6 marks. That's an amazing 3% of your whole GCSE, so it's really worth knowing how to answer these kinds of questions. Okay, so what kind of thing were we talking about? Well, here's an example question. It says, describe an investigation, there's those magic 3 words, to find out about the most suitable amount of calcium ions needed to get the best growth in rats. So we've got describe an investigation, and it's a 6 marker. And so we're going to utilize this CORMMSS mnemonic to help us structure our answer. So what do these letters stand for? Well, C, the first one, is in fact conditions. So the conditions are really the different situations that you're going to put your organism through. So we'll look at some examples of that in a second, because that's sometimes a little bit hard for people to understand in the first instance, but it might help you for now to think that this is the way that you're going to change your independent variable. The second letter, O, stands for organism, and we're looking at the ways in which the organism has to be the same. So that might be that they should all be the same species, or they should be the same age, or perhaps the same sex, or maybe the same starting size. R stands for repeat, so you're always going to say that you're going to repeat your experiment, and ideally you're going to say how you're going to repeat it and what ways it's going to be repeated. Both of the M's stand for the same word. They stand for measurement. So the first one is the measurement of the dependent variable. So what exactly are you going to be measuring in your experiment? Again, we'll look at some examples of this in a second to make that a bit clearer. And then the second M is for the measurement of time. So how long are you going to take your measurements for? Is it going to be 10 minutes, 10 years, 10 days? What's it going to be? It's probably worth saying that in some mark schemes you'll see the M marks divided up a little bit differently than this, but if you do both of those things, you can always be guaranteed you'll get the marks. The S's both also stand for the same thing as well, and they stand for standard, so standard 1 and standard 2, and really this means what things are you going to keep the same in your experiment to ensure that it's valid. And we'll think about what kinds of things they might be in just a sec. Okay, so let's go through a worked example. So here's a question again. Describe an investigation to find out the most suitable amount of calcium ions needed to get the best growth in rats. It'll be helpful for you to think first, what's my independent variable and what's my dependent variable here? So independent variable, remember, means the thing that I'm changing, the thing that I think is going to cause a difference. So if I look at that question there, well, I can see that the thing that I think is going to affect the growth of the rats is the amount of calcium ions. So that's my independent variable. And then the dependent variable is the thing that I'm going to measure, the thing that I think is affected by something else, the thing that I think depends on something else. And that thing is going to be the growth in rats. The growth in rats depends on the amount of calcium ions. Okay, so now I've got those sorted, I can write down CORMMSS on my exam paper and can start sort of scribbling a little plan to think about how I'm going to answer this question. Really is worth spending just a minute or two planning an answer like this. So the CORMMSS begins with C, and C stands for, can you remember? Stands for conditions. So here, the conditions, the different situations that the rats will be in, is simply different amounts of calcium in their diet. So you could just say different amounts of calcium, you could describe a whole range of calcium, but in some way you're going to say that the rats should be divided into 2 or more different conditions in which they have different amounts of calcium. And then the second letter is O. What did that stand for? So O stands for organism, and the way that they're the same. So you'll see that this says same age, same strain, so that means type, or same species, same gender. And these O ones are almost always going to be the same kinds of things. If you think I'm going to pick from the list same species, same age, same sex, and same gender, and you pick the 2 that you think are most appropriate here, then you're pretty likely to get that O mark. So the way in which the organism is the same. The R mark is super easy. You just got to say that you're going to repeat it, and here you're going to say, oh, I'm going to repeat it with several different rats, or I'm going to repeat it with several different groups, something like that. So not just the word repeat. Sometimes that won't get you the mark. You just got to say a little bit more. In what way is it repeated? Okay, and then the first M, remembering the M stood for measurement. The first M is our measurement of the dependent variable. So we said that the dependent variable was the growth in rats. That's the thing that depends on something else. And how are we going to measure how much the rats have grown? Well, let's measure their mass. There are a few other things that you potentially could have done to measure the growth in the rats. Sometimes people tell me for this question that they think they'd like to measure the length of the rats or something like that. That's okay, but in general, if you're going to measure how much something's growing, we like to use mass for that. The second M, remember, is the period of time that we're going to take our measurement for. And you can really just pick anything that you think is sensible. So here they're suggesting that a sensible amount of time would be 1 day or more. It has to be enough time for you to see a change in the dependent variable. So obviously our rats aren't going to grow much in 30 seconds, but our rats will be dead if we've left them for 30 years. So a day or more is about right. S, coming on to now. Can you remember what that stood for? That stands for standards, so the things that we're keeping the same. So not things about the organism, because we've already dealt with that in O, but other things in the experiment that need to be kept the same. So for this question, the options were they should be given the same mass of food. So it wouldn't be fair to give them different amounts of food, because obviously that's also going to affect their growth. They should be given the same type of food, they should be given the same diet, they should be given the same amount of water, they should be kept in the same cage, and they should be kept at the same temperature. And you'll notice that these S things are all things that would affect the dependent variable other than the independent variable. Okay, another worked example. So here the question is, commercial flower growers keep their plants in greenhouses under artificial light. Describe an investigation, remember those 3 words that tell us it's a CORMMSS question, to find out if the length of time that plants are exposed to light affects how long it takes for flowers to appear. Okay, so what's our independent, what's our dependent variable? So the independent variable here is the length of time that plants are exposed to light, because that's the thing that we're going to be changing, and the dependent variable is going to be how long it takes for flowers to appear, because that's the thing that we think depends on something else. We think it depends on how long they're exposed to light for. Okay, so we know it's a CORMMSS question because it said describe an investigation. So we write down our CORMMSS, and we start to make some notes about what we're going to write. So for the C, we're looking for the conditions, so the different conditions that our flowers are going to be exposed to. So since our independent variable was the length of time plants are exposed to light, then our conditions are simply having the plants in different groups where they're exposed to different amounts of light. You were allowed, you'll see here, to say have one of the groups exposed to light and one to dark, but you weren't allowed to say different light intensities because that's something slightly different. We're talking here about how long they're in the light for. So you could say something along the lines of, I would set up the experiment with two groups of plants, and the two groups will be exposed to light for different amounts of time. Okay, on to O now. O stands for organism and the way that they're the same. Remember, again, here we're seeing same species, same variety, which is like the same type, same age, and we said before we could also potentially have talked about same sex and other things of that sort, like the same size. Same sex isn't particularly relevant here because we don't think that that's likely to affect things, and sex in plants works a bit differently anyhow. So then the R stands for repeat, and here you were allowed to just simply say the word repeat, but you could have said in what way you're going to repeat it if you wanted. M stands for measurement, and the first measurement is the measurement of the dependent variable. The dependent variable here, remember, was how long it takes for flowers to appear. So we're going to measure that simply by recording the time it took for them to produce flowers. We could potentially have given them a set length of time and seen how many flowers they produced in that set amount of time. But we're essentially just going to be looking at how are we going to measure the dependent variable. What exactly are we going to take a measurement of? We're going to take a measurement of time. If you wanted, you could even say the apparatus you'd use to do that. So a stopwatch or a clock of some sort, a timer of some sort. The second M, remember, was for the length of time that you're going to do your experiment for. So here it suggests maybe we're going to use days or weeks or month, and again, look, they're there referring to the use of the apparatus, a watch or a clock or something. S stands for standards, so it's the things that you're going to keep the same other than the things that are the same about the species, and the things that you think would also affect the dependent variable. So here, temperature, light intensity, carbon dioxide, water, humidity, minerals, nutrients, the soil, pH, lots of different options, all other things that might affect how long it takes for flowers to appear. Okay, our third and final worked example, and this one's a little bit different. So it's a slightly longer question, it's a really off-putting big length of text, and it starts to produce large amounts of human insulin that genetically modified bacteria are grown in a fermenter. Describe an investigation, so our three words are again there, to find out if temperature affects the amount of insulin made by genetically modified bacteria. Your answer should include experimental details and be written in full sentences and paragraphs. And this is a good point to point out that you can't just write the letter CORMMSS on your exam paper, but you've got to write this as full sentences. Just use CORMMSS for your planning. Okay, so let's shorten that out a little bit, and we'll just call that describe an investigation to find out if temperature affects the amount of insulin made by genetically modified bacteria. Okay, so first thing, identify what your independent and dependent variables are. So the independent variable here is temperature, and the dependent variable is the amount of insulin made by genetically modified bacteria. So note down CORMMSS, remember what each of the different parts of CORMMSS stand for. Maybe pause the video at this point and see if you can write an answer to this question using CORMMSS. And then once you've written it, unpause it and we'll see how you got on. Okay, so the C in CORMMSS stands for conditions. So the different conditions here are simply different temperatures, different ranges of temperatures. So we're going to say something like, I'd set up the experiment and I'd have different groups of bacteria that were exposed to different temperatures, or you could state specifically what range of temperatures if you wanted. The O is the way in which the organism is the same. So we've got to think, well, what is the organism here? It's the genetically modified bacteria, that's the thing that's alive. And again, same kinds of things. Same species, same mass, same type of bacteria, and here they're talking about same amount, number, etc. So remember, we're picking the most appropriate from that list that we've gone through before. R stands for repeat, so we're just going to simply repeat. And then the M's here are actually slightly different to how we've talked about them before. So we've talked before about putting down our M for the first one as being a description of the dependent variable and how we're going to measure it, and the second one as being a measurement of time. So if we'd put both of those things for this question, that would be correct, and we definitely get the marks, because here the M is actually split into two parts. So what we normally put for our M1, here you get essentially 2 marks for. So the first part is to say that you're going to measure the amount of insulin, and then the second part is for saying, well, how are you going to measure the amount of insulin? So what is the sort of the nature of the quantity? So we're going to measure the concentration of insulin, or the mass of insulin, or the volume of insulin. So you see that occasionally these M's are slightly different, and so that means that it's good practice for us when we're answering CORMMSS questions in the M1 to say, when we're talking about how we're going to measure the dependent variable, what are we going to measure, what sort of quantity of it, is it mass or volume or what have you, and also to say what apparatus we're going to use, because sometimes that carries a mark. And then the standards here is the same as they ever are, so the different options were same pH, same food, same oxygen, same time period, same type of fermenter, keeping it sterile, so on and so forth. So again, you see that these are all things that could potentially also affect the amount of insulin made. You'll also notice, I'm sure, that one of the standard marks is for saying having the same time period. So again, if you were doing our normal method of using M2 as being stating the time period, you'd potentially get that standard mark for that. Okay, so just to quickly refresh our memories of what each of those letters stood for, maybe pause the video now and see whether you can remember what each of these letters stand for. So C is conditions, O is the organism, R is repeat, M is for measurement of the dependent variable, and then the second M is for the measurement of the time period, and then the S's are for the standards, the things we're keeping the same. Okay, so in the description I will put a couple of written model answers to these questions. Please do feel free to comment with your answers, and I'll try and reply to you and give you some feedback on them. And do remember to subscribe to the channel for some more videos that will be coming up looking at these kinds of things.